Hi everyone, my name is Soto, and today I will be presenting Epitope-Evaluator, an interactive web application to study predicted T-cell epitopes. The epitopes are small peptides within proteins that are able to trigger immune responses, in particular the T-cell epitopes that can be recognized by MHC alleles, can activate cytotoxic and helper T-cell. Nowadays, there are many software to predict T-cell epitopes, but there are few software to do the analysis. Important features of these epitopes include the promiscuity, the conservation, and the location. The promiscuity is the ability of the epitope to bind to several alleles. The conservation helps to identify epitopes that can generate protection against different pathogen strains. And the location is important to correlate the number of epitopes with functional domains or non-toxic domains. So to help with this analysis, we have developed Epitope-Evaluator. The workflow starts with a prediction of epitopes, so the user needs to upload the FASTA file and the alleles ID. However, for the Epitope-Evaluator, we need to upload the prediction file and the FASTA file. This is the website that is available at this link, and also the code can be downloaded from the GitHub. Here we have 5 different sections. The first section is the title section, where there is the home page, the about page, the run analysis, the documentation, and the tutorial. Then we have the tools section, where there are 6 available tools, the parameter sections that you can set before running each analysis, the help sections that details all the parameters and the meaning, and the output sections that gives all the interactive plot and the tables that can be downloaded. Among the different tools, we have the epitope distribution that is important to identify alleles that recognize fewer epitopes, for example, the epitope intersection that shows you the number and the sequences of the epitopes predicted to bind to a particular combination of alleles, the epitope density that correlate the length of the protein versus the number of epitopes, the viewer to see the location of the epitopes in the protein, and also highlighting which of them are more promiscuous than others. Then we have the promiscuity, important to identify a strong binder but also weak binder epitopes, and the conservation to see which epitopes are conserved and which are specific to different variants or to different strains. So Epitope-Evaluator is very important in vaccine design. For example, we can see that in a fields approach, it can help you to identify highly immunogenic proteins that can be postulated as whole protein recombinant vaccines. Also, in a second approach, we can use it to identify domains enriched of epitopes to use as subunit vaccines. And lastly, it can help to filter promiscuous epitopes in order to assembly in a multi-epitope vaccine. Moreover, we use Epitope-Evaluator to study immune responses based on the binding of the epitopes. Here, we predicted epitopes from the SARS-CoV-2 proteome. We have the class 1 epitopes and the class 2 epitopes. We identified 3 alleles that recognize fewer epitopes and that can be correlated with a more severe infection process in the patients. We also identify highly antigenic proteins. For example, in addition to the spike, we identify the nucleocapsid, the ORF3, and the membrane proteins that can be used as complementary vaccines because they have several epitopes for class 1 alleles and also for class 2 alleles. And finally, we studied the variability of the class 2 epitopes within spike variants. We focused on the Delta, Wuhan, and Omicron variant. We identified 200 of epitopes that are very conserved, but also the epitopes that are specific to Omicron and specific to Delta. These results correlate with the location of the different mutations. So as Omicron has many mutations, there are more variability in the formation of new epitopes. So in conclusion, Epitope-Evaluator is a new software very important to study epitopes, vaccine development, but also to study immune responses. Thank you so much.
Excellent presentation, Luis. Please, if you have any questions, ask for them in the Q&A section. Luis, I want to make one question to open with. Do you have any further experiments planned to continue this project?
Yeah, definitely. There are a couple of things that we can continue with this work. One of these about integrating with gene expression, because maybe there are some proteins that are more expressed than others, and that they can be more important than maybe epitopes located in a very low expressed proteins. This is one thing. And the second thing is integrating with B cell epitopes. So not only focusing on T cell, but also with B cell epitopes that can be used for the recognition of antibodies.
Excellent. We have a question from Alatanov.
Yeah. Oh, sorry, I didn't realize I was muted again. Yeah, thanks. That was a great talk. I was just going to say that, yeah, it's great that your web tool can identify those epitopes and the likes, and you have a lot of them. So I was just thinking that the reviewers will always want to be sure that you can do some kind of follow-up experimentation to validate all these predictions. Otherwise, they would even tell you that yeah, they think you have some insights, but especially if you're trying to push it to very high tier journal, they would usually not accept it unless you can do that follow-up experimentation. So I was wondering if you have plans for stuff like that.
Yeah, definitely. There are two main points here. The first one is the Epitope-Evaluator is more about a tool to analyze predicted epitopes. So they are using prediction from different software that are very well supported. And the second point is that we are working in a development of a vaccine against Clostridium perfringens. So one of our next steps is the validation of this vaccine. But that is right, your comment.
Okay,
Thank you.
thank